No. Yeah, it's not. Oh, okay. Oh yeah. We do have to see video. Her app is office out there actually. That's just the like without moving over. Yeah.
Okay, let's get started. I know busy week for a lot of you, so appreciate you coming. We were working through chapter 7, which is all about growth. We talked about the sort of things that microbes need, the elements that they need to grow. We could describe what growth looks like in bacteria, exponential growth. We talked through a problem to kind of taking into account generation time and to work out microbial numbers at a later time or a certain number of generations. And we talked about how to count different ways to count microbes. And then we started talking about different environmental conditions that can impact growth, things like oxygen requirements, temperature, pH, things like that. So I almost worked our way through this, and we ended last time talking about the physical effects of water. So osmotic pressure. And when we left off last time, I was talking about how osmotic pressure, that really refers to whether bacteria, high osmotic pressure refers to a hypertonic environment. So I don't know if I said that quite that way, but high osmotic pressure, again, that we equate that with an increasingly more and more hypertonic solution. So high solutes in the solution that the cell is in. So usually that's bad for cells because usually they're going to dehydrate, but there are organisms that are specifically adapted to actually grow best, or they prefer to grow in solutions of high solutes. So we call those organisms osmophiles. Okay, that means they grow best in high osmotic pressure. So they have ways to keep their cells from losing water. Sometimes they concentrate solutes inside of the cell so that it's not such a difference outside of the cell and inside of the cell. And then halophiles are osmophiles that specifically grow best in high concentrations of salt. So grow best in increased concentrations of salt. Okay, so again, it's a specific kind of osmophile. There could be organisms that grow best in, say, a high concentration of sugar or some other solute other than salt. So this picture, it's not obvious what this picture is. This is from your textbook. And this is an aerial, probably a satellite photo of the Great Salt Lake. So ocean water has a salt concentration of about 3%. And the Great Salt Lake is anywhere from between 5% salinity to 27%. So 27%. The Dead Sea over in the Middle East is about 10 times saltier than oceans, about 30% salinity. Okay, so really it's only microbes that can grow in that high salt concentration. So halophiles, these are pretty extreme organisms. They're in that archaea domain, not organisms that you're going to come in contact with every day. But apparently, you know, the color of the water there is changed. It's kind of purplish. It's hard to see that because of all the bacteria in the water. Okay. Oh. Thanks. All right, and the last thing, just to kind of close out this chapter, I don't think they talk about this in your book, but if we're talking about kind of the environment that bacteria specifically are in, we have to remember that they may act a little bit differently in the environment than what they do in the lab. We have them by themselves in pure culture, just because of the other organisms that they're around. So we have terms to try to describe the relationship that those organisms have with other species. And I should say this is what makes it so difficult to kind of understand what's going on in our microbiome in our bodies, because we know, some of the organisms that are there in our large intestine, for example, and we can grow them up in the lab, but when we grow them in the lab by themselves, they act differently than if they're with those hundreds of other species. So to try to figure out, what organisms need to be there to make us healthier, which ones are making us sick, it's not an easy task. So, all right, so some of these relationships, antagonistic relationships, that just means when you have two organisms, one organism benefits and one is harmed. Okay, you could also think about this as parasitism. Okay, that's an antagonistic relationship. You can think of lots of examples of this. Certainly, there are organisms in our body that are harming us. We're giving them all these nutrients and they make us sick. Here's an example. This top picture here, that big white spot is some mold that's growing on a plate of some bacteria that's streaked out there. And if you look closely, there's kind of a zone. There's kind of this zone around the mold where there's no bacteria growing. Okay, that mold is secreting something that's antagonistic to the growth of that bacteria, probably some antibiotic. Okay, a synergistic relationship is a little bit harder to think of examples of that. That's where maybe two members can live separately, but benefit from the association. And again, it's a little bit harder in the microbe world to kind of think of examples of that. You could say bacteria that live in biofilms. Okay, they're a little harder to kill when they're in biofilms, so they can live separately, but sometimes it's advantageous for them to live in, great big mats of bacteria. There's lots of examples, though, of symbolic relationships. That just means that both organisms benefit. And in fact, so much so that one organism is rarely found without the other. Okay, so lots of examples. This picture here shows a particular kind of bacteria in the genus Rhizobium that lives in association with the roots of pea plants. Okay, so the bacteria fixes the nitrogen in a usable form so that the pea plant can grow, and in return, the pea plant supplies energy to the bacteria. There's bacteria in the rumens of cow's stomach. Cows could not digest grass without the bacteria in their rumen. So again, the organism is getting all the nutrients from the cow, and the cow in turn is able to digest the grass thanks to the bacteria. Same thing, bacteria in the guts of termites. Termites couldn't digest that wood without, it's actually the bacteria that do that for them. Okay. So let's see, let's do a practice question. I think probably one of the more difficult things that we waded through last time was talking about all those toxic forms of oxygen. So I'll give you a practice question there. Give you a minute to read that and think about what you would answer. Okay, so hopefully you recognize those three enzymes are ones that we said that organisms need to have some combination of them in order to detoxify some of those toxic forms of oxygen, like superoxide or peroxide, hydroxyl or peroxide. Yeah. So if an organism doesn't have those, then it's not going to be able to live in atmospheric oxygen. So it's going to be an obligate anaerobe. Okay. Hopefully, so you came to that. This alkalinophile, that doesn't have anything to do with it. Toxic oxygen, of course, that's an organism that lives in alkaline pH. Questions there? Yeah, okay. So anaerobe means without oxygen. Okay, so if an organism's a strict anaerobe, then they're poisoned by those toxic forms of oxygen. They don't have any metabolism that takes advantage of oxygen. So we're going to talk about metabolism next and the role that it plays. So, okay. So moving on, chapter 9. All right, so let's see, we've been through chapter 5, the eukaryotes, chapter 7, growth. So there's homework that you can be working on this weekend. We'll spend about a lecture and a half probably here on metabolism. And I talk about metabolism topics a lot with my husband. He's a biochemist. And some of the things I was talking about with him, he said, you know, I was trying to tell him the level that we're teaching metabolism. And he said, you know, some things it's hard to do just a little bit, right? Like metabolism is really complicated. You understand it so much better when you look at structures and then what the enzymes do to change each structure. But we're not going to do that. We're going to try to do metabolism just a little bit. And sometimes that presents problems, but we're really going to look at it kind of an overview. Yeah, we're going to look at pathways, but we're not going to be too concerned with intermediates. Think about kind of what we start with and what we end with. So we're going to little review of metabolism and the components of metabolism, like redox reactions, ATP, electron carriers, that kind of stuff. We're going to walk through some pathways that primarily will break down sugars, but we'll talk about what happens with, you know, proteins or lipids when they're broken down as well. So we'll talk first about respiration, both aerobic respiration and anaerobic, how bacteria and archaea can respire with something else as an electron acceptor other than oxygen. And then fermentation, how that's different than anaerobic respiration. Okay, so quite a few things to walk through. Don't be afraid to stop and ask questions or slow me down if I blow through something that didn't make sense. So, okay. So metabolism, we could think about that. Metabolism is just the chemical reactions in a cell that will produce ATP or produce precursor molecules that a cell needs to grow. We talk about those reactions mostly as being either anabolic or catabolic. So an anabolic reaction, hopefully you've heard those terms before. These are reactions that build molecules. Okay, in other words, they're biosynthetic anabolic reactions. And they're going to be endergonic. In other words, you have to have an input of ATP for those molecules to be synthesized. Okay, On the other hand, a catabolic reaction is going to be a reaction or reactions that break down molecules. Okay, and that's exergonic, so we gain ATP from those reactions. And those have to be pretty tightly balanced within a cell. You can't have a bunch of anabolic reactions without having a catabolic reaction to provide the energy for that. Now, actually, a lot of the reactions, we'll throw one more term in there, are actually considered amphibolic. Remember that prefix amphi means both. Okay, amphitrachus flagella is a flagella at each end of the cell. Amphibian, both water and land. So amphibolic, these are reactions that can proceed in either direction. So I'll try to kind of note that as we go along. Actually, a lot of these processes that we're going to talk about, the reaction can go one way, or with just a few differences, it can proceed back the other way as well. Okay, and just every once in a while I run across a mnemonic, but I think, oh, maybe that will be helpful. If this mnemonic, ABCD, anabolism, building molecules, catabolism, degradation. I don't know. That may be helpful. Okay. All right. So just kind of again, putting a picture to it there. When a cell, gains nutrients, then by catabolism, those nutrients are broken down. ATP is released, and we get some precursor molecules. Those precursor molecules are used in anabolic reactions to make cell structures, things like membranes or ribosomes or whatever, or maybe stored as energy. Okay, both catabolic and anabolic reactions in the cell, they're not perfect. They're going to lose a little bit of that energy as heat, kind of inevitably. All right, but again, tightly balanced, both catabolic and anabolic reactions. Okay, redox reactions. So when you have an oxidation reaction, okay, then if say this molecule is oxidized, that means that they are losing an electron. Okay, so that oil, you've heard that mnemonic, oil rig, probably. So oil rig, oxidation is loss of electrons. When a molecule gains an electron, okay, then that's called a reduction. I know that those terms, that doesn't really make a lot of sense, but reduction is gain of electrons. Okay. You could think about the net negative charge is reduced. Okay, so if it's gaining an electron, it's getting less negative, I guess, more negative, reduced. There is, oh, there it is right there. So this YouTube link here is it's like a 10-minute YouTube video from Crash Course Chemistry, and it's actually really funny. And the first few minutes of it just kind of talk about how these are bad names, oxidation, reduction. They were used to describe these reactions before we really understood what was going on. But some names you just kind of stick with. He also talks about how they're really, really important. Like you just got to know this stuff sometimes, and that it's in metabolism, it's really all about the electrons. So we're going to see about how those electrons that are pulled off reactions are converted into a huge amount of ATP. So it's important to follow the electrons, I think one of the very confusing things about redox reactions, and they do go into that video how to assign oxidation states and stuff that we're not going to worry about. But I think one of the confusing things is, and that we often forget, is that protons often accompany electrons. And we don't often write the electrons into some kind of an equation. So for instance, a hydrogen atom, remember, is equal to a proton and an electron. Okay, so when you see those protons in a chemical reaction, just remember there's an electron with it as well. So when some molecule is gaining a proton, it's also gaining an electron, even though, again, you don't really see that in the reaction. Okay, so let me illustrate that as we talk about our electron carriers and show you some reactions here. Electrons in the cell, we don't let them just kind of go wherever they want because they will cause a lot of damage just loose in a cell. They have to be chaperoned, to think about it that way. So electron carriers like NAD, FAD, NADP, they are the chaperones of electrons. Okay, so nicotinamide adenine dinucleotide, this is that carrier in its oxidized state. Okay, and it can actually hold not just one electron, but it actually can accept a hydride ion, which is 2 electrons and a proton. Okay, so when it's reduced, we write it as NADH plus H plus. Okay, so here, what we don't show here, it's actually holding a hydride ion, which is 2 electrons and a proton. Okay, so that's why there's the H plus left over. Again, 2 electrons and 1 proton is carried by that NADH and escorted around the cell. FAD can actually hold 2 protons. So when it's really reduced, it's FADH2. Okay, and both of these electron carriers are going to shuttle those electrons to the electron transport chain. Okay, so that's kind of the last step of aerobic or anaerobic respiration. So we'll talk much more about that. NADP is a carrier that also accepts hydride ions. There's a phosphate on there as well. So its reduced state is NADPH plus H plus. Now those electrons, though, don't go to the electron transport chain. They are used in anabolic reactions. Okay, so those electrons don't get converted into ATP, but they are helpful and necessary for the cell to make cellular structures. Okay. And remember, anytime, I can't remember if I said this, but anytime you have an oxidation, you have to have a reduction at the same time. You can't have one without the other. So as we'll see, when these carriers, when they get reduced, they have to have somewhere to dump off their electrons. Otherwise, they can't cycle back to their oxidized form. Okay, so we'll see how important that is. What if they can't dump them off? Then the whole pathways are going to stop. Okay, to keep the pathways going. We got to mention ATP. Okay, this is the energy currency of the cell. Structure there, adenosine, which is the adenine base, which we're going to look at when we look at our DNA and RNA pretty soon, ribose sugar, and then 3 phosphates. Those bonds between the phosphates there are high energy phosphate bonds. So when we break one of those bonds, that energy can be used to do work in the cell. Okay, so when you break one of those bonds, then you're left with ADP, and that can cycle back, get another phosphate added to it, either by some chemical energy or energy from the sun. Okay. Now, there's 3 different ways that a phosphate can be added to ADP. We call that a phosphorylation, when a phosphate is added to make ATP. Okay, so let's talk about those 3 different ways. We're really only going to mention 2 of them moving forward, but we'll at least talk about all 3 of them. Okay, the first is called a substrate level phosphorylation. Okay, now we're going to look more at glycolysis later, so don't get wrapped up in the pathway just yet. But in that pathway, kind of down the lower part of the pathway here, and this is rather small. I'll just blow it up a little bit bigger here. But you can see on this molecule here, 1,3-biphosphooglycerate, it's got 2 phosphates on it. And when one of those phosphates is pulled off of the substrates and added to ADP, then we get ATP. Okay, same thing down here. This phosphate is pulled off of phosphoenolpyruvate and added to ADP to make ATP. Okay, so usually that phosphate that is used is from substrate and metabolism. Occasionally it can be just in solution somewhere. It's not always obvious, but we get substrate level phosphorylations both in glycolysis and the Krebs cycle. The other way that ADP can be phosphorylated to ATP that we're going to talk more about, and this will make more sense later, is an oxidative phosphorylation. Everybody sort of assumes that this must require oxygen. That actually is not the case. This is when NADH is oxidized back to NAD. Okay, so when the NADH is dropping off its 2 electrons and it's a proton at actually the electron transport chain and oxidized back to NAD, that starts this cascade of reactions that results in lots of ADP being phosphorylated to ATP. Okay, so it's that oxidation of NADH actually. Okay, and that again is going to occur at the cell membrane of prokaryotes in the electron transport chain. So, okay. The other way that ADP can become ATP is a photophosphorylation that happens with organisms that are photosynthetic. So the energy of the sun catalyzes that, and we're mostly interested in organisms that are causing disease, and no disease-causing organisms are photosynthetic. So we're not really going to talk about photosynthesis, so that's the only time I'll mention it here. Okay, so we'll talk about those terms again later. Also want to review a little bit about enzymes. Okay, so a little terminology. You know what an enzyme is? An enzyme is going to speed up some reaction. Okay, the way it's often described is that enzymes lower the activation energy necessary for a reaction to take place. Some of these reactions would take place anyway in the cell, but they would take place so slowly that it wouldn't support life. So speeds up reactions. Enzymes, if you see a molecule that ends in ASE, then you know that it's an enzyme. Beta galactosidase, that's an enzyme. There are other enzymes that don't end in ASE, and then unfortunately they don't follow the rule. If it ends in OSE, then it's a sugar, right? So that's different. All right, so enzymes speed up reactions. Enzymes are going to work on substrates. Okay, usually you write the name of the enzyme over the arrow, and then the enzymes turns the substrate into a product. In this picture here, they call the substrate reactants. Okay, same thing. We're gonna study in lab in the next couple weeks, lots and lots, well, several different enzymes, and trying to understand if certain organisms can produce certain enzymes. How do you know if an organism can make an enzyme? Well, you look and see if some product shows up. You can test for the presence of that product, or maybe you might see if the substrate disappears. If either one of those two things happen, then the organism must be able to make that enzyme. Some enzymes are simple. Okay, and a simple enzyme is just going to be protein. Okay, but there are other enzymes that are more complicated in that we would call it a conjugated enzyme that needs some non-protein component to function. Okay, so a lot of enzymes need either a cofactor or a coenzyme. Oops. In order to function. So a cofactor is usually some kind of inorganic ion. It's actually written right there, an inorganic cofactor. That's necessary for usually the shape of the active site to be the right shape. And a coenzyme is just a cofactor that's organic. Remember, it's got carbon and hydrogen bonds. Okay, yeah, question.
What is so the conjugated enzyme is? It seems that
These non-protein components of function. No. Okay, so here's an example of a conjugate enzyme. The protein part of the enzyme is called the apoenzyme, and then the whole thing together, along with any coenzymes or cofactors that are present, is called the holoenzyme. Okay, and the part of the enzyme where the substrate binds, that's known as the active site. Okay, so you can see here in this example, without the cofactor and the coenzyme, the active site would be a very different shape. Substrates wouldn't be able to bind. Cofactors are often some inorganic ion, something like copper or zinc or iron, for example. And coenzymes often are some kind of vitamin. All right, so just some terms to be familiar with. Remember that enzymes, basically there's that lock and key hypothesis, how here's the enzyme and the substrate has to fit kind of just right. There's actually a little induced fit there, but that has to fit, pretty close to being just right. The enzyme is not changed or used up in the reaction at all. It will just change the substrate. In this case, it cut the substrate into two different products. Okay, and then the enzyme can go and do the same thing again. And this is just the figure from your book that shows how, the cofactor or the coenzyme has to be there, in this case, in order for the substrate to fit right. Enzyme activity. So, of course, there's lots of different enzymes that are going to need different physical parameters, but enzymes will have some ideal temperature where they will catalyze reactions the quickest. And oftentimes, soon after that ideal temperature is met, Soon after that, you get a little bit hotter than that, and the enzyme will denature and not be functional at all. So just like cells have an ideal temperature, it's because the enzymes in cells have an ideal temperature that they're going to be working and growing the quickest. Same thing with pH, fairly narrow pH range. This shows an ideal pH of 7, but again, it could be different depending on what enzyme we're talking about. And we have to think about the substrate concentration as well as the enzyme concentration. If you have a certain number of enzymes, as you get more substrate molecules, then that enzyme is going to be able to work faster because there's more substrate available to make more product, but only to a certain point. If you get more substrate than enzyme, then that's gonna, the rate is going to level off. Okay, same thing if you have a certain number of substrate, make more and more enzyme, the reaction will move quicker, only up to a certain point. Let's see, cells typically want to maintain kind of a tight control on their metabolism, so they will usually be able to have some things built in that will regulate how quickly enzymes work and whether they work at all. Okay, so couple different kinds of regulation. A competitive inhibition of an enzyme is where you have some molecule that fits right into the active site of the enzyme. Okay, so then it blocks the substrate from binding. You don't get any product. An allosteric, or sometimes we refer to it as non-competitive inhibition, is when there's some molecule binds to an allosteric site, or you could think of that as other, okay, some other site other than the active site, and that changes the shape of the active site, even though it's not right on the active site. So in this case, the substrate can't bind. Again, both are just ways for the cell to kind of, or sometimes it's something from outside of the cell that keeps those enzymes from working correctly. That non-competitive inhibition actually can be both, maybe a better term for that is non-competitive control, because it can work both ways. Like we just said, you can have an allosteric inhibitor that keeps the substrate from binding, but in some cases, there has to be some kind of allosteric molecule that binds in order to change the active site, so now the substrate can bind. And without that molecule, then the substrate can't bind. So it's kind of more of a positive control or a negative control, and that's not always inhibition. And of course, we know instances where there's some kind of feedback inhibition. Usually this is in some metabolic pathway. Here you've got the first enzyme in the pathway, a substrate binds to that, and then there's maybe a few intermediates. You end up with this end product. Well, if the cell has a whole bunch of that, maybe it makes sense that the cell doesn't keep producing more of it. So that end product can actually bind allosterically, almost always going to be allosterically. It's not going to have probably the same shape as the active site. And that will inhibit that pathway. Okay, so the cell has a bunch of it that doesn't want to make some more. We'll see in a few weeks how the cell can actually, a different kind of level of control, keep from producing the enzyme in the first place. That's kind of the level of transcription. We'll talk more about that as we move forward. Okay, all right, so let's do a question before we jump into some pathways. Take a minute, read that carefully. See if you and the person next to you think it's the same answer. It's one of those wordier questions you just kind of have to slog through and read it all kind of carefully. Correct answer is A. All right. Okay, so if there was a cofactor and that along with the apo enzyme, that's actually part of the apo so sorry, the apo enzyme is the protein portion, along with any cofactor or coenzyme that would make up the hollow enzyme. So really, either that one's not true. We talked about how if you don't have enough enzymes work on substrate, then that's not necessarily going to be right. Anytime you see always or none, that's kind of a tip off that might not be the right answer if you're looking for something that's true. Substrates bind to the active site, not the allosteric site. And this one, that sounds so true, but I just made that up. So, okay. All right. Coenzymes are just the organic cofactor, non-protein portion that has to be there for some enzymes to work. Okay. So as we get into some pathways, things I'm going to pay particular attention to, and I tried to kind of make these things clear on the objectives, as clear as I can, kind of what level that we're going to try to learn these things. We're going to pay particular attention to whether pathway, you have to put in some ATP and whether you get ATP out or not. There's going to be some key juncture points as to kind of which direction metabolism can go, depending on what a cell needs. We're going to pay attention to what we start with in a pathway and what we get out of it, especially are there, electrons maybe, follow the electrons, that's going to be important. Counting carbons can help you from getting confused as well. So again, no structures, no particular enzymes. I will mention some intermediates and pathways, but again, those are the intermediates are not something that you're going to need to memorize as well. So we're also paying particular attention to things that are uniquely microbial. And I'll try to compare, like, okay, this is how it happens in eukaryotic cells, and this is how it's different in prokaryotic cells, mostly just where it takes place in the cell. All right, so I kind of like this summary slide that our old textbook has had. I'll show that probably several times. A summary of glucose catabolism. So usually when we're talking about metabolism, we're using glucose, because that's the most readily available thing that cells usually have. It's usually going to be the first choice of what they're going to use. A lot of cells can use other things like proteins and lipids, and we'll talk about that a little bit, but just kind of by default, if glucose is there, that's what they're going to use. And you'll notice that this diagram has a green portion and an orange portion. Okay, so the green portion is respiration pathways, and the orange portion is fermentation. Okay, so glycolysis, that's the first step in both respiration and fermentation. Okay, so that is going to be the first thing that we'll start with. We'll come back to fermentation and focus first on respiration. So glycolysis, that's the splitting of sugar. We end up with a couple of pyruvic acids, and those will, with a little transition step, get fed into the Krebs cycle. You can see places where there's electrons coming off NADHs, there's some FADHs coming off the Krebs cycle, spots where ATP is produced, but those electrons are going to come off both of glycolysis and the Krebs cycle and get fed into some kind of electron transport chain where we have some final electron acceptor. We're used to calling that oxygen, but and all we can use is oxygen, but a lot of bacteria can use a lot of different things, we'll say for now, or something else, okay, for bacteria. So it's a very similar respiratory pathway if something else is used, like nitrate or sulfate as the final electron acceptor. A few changes, but mostly the same pathway. Okay. So let's start with glycolysis. And this figure that your book gives you for glycolysis, this is just about as simplified as we could possibly make it. I call it like the third grade version of glycolysis. So we'll start with that. We'll get a little bit more complicated, but not too much. So kind of overview. Okay, so first of all, I called this glycolysis. Really, a more correct name for this is the Embden-Meyerhof-Parnas pathway, and that's written over here. But let's write it again. Okay. Again, usually we just refer to this as glycolysis. Glycolysis is a great name because it describes what's going on, the splitting of sugars. But it turns out there are a few other glycolytic pathways where sugar is split. Okay, so we would more correctly call this Embden-Meyerhof-Parnas glycolytic pathway. Okay, or EMP, right? You see that abbreviation. Okay, you have to recognize that name. I've read papers where they talk about EMP pathway. I'm like, oh, wait a minute, what are they talking about exactly? Okay, so that's just the best name for it. And overall, what happens here, right? We're starting with that 6-carbon glucose. We have to input some energy to get things started. This fructose diphosphate, that is actually not a symmetric molecule, so it doesn't split. This is an oversimplification into two of the exact same things. I just want to mention that the next figure at least points that out. We'll come back to that. But then we get 2-glyceraldehyde 3-phosphates, and then the lower portion where we get the energy payoff, or we're getting energy out of it, is at the bottom here. So we get some electrons carried by NAD and some ATPs. So if we think about our net gains for glycolysis, okay, we're getting 2 NADHs. We put in 2 ATP and we got out 4. So we're net gaining 2 ATPs. And of course, we're getting 2 pyruvates, or you can call that pyruvic acid, same thing. Okay, where is this taking place in the cell? Cytoplasm, right. So for both prokaryotes and eukaryotes, that's going to be in the cytoplasm of the cell. All right, and who is doing glycolysis? What kind of organisms? Pretty much everybody. All right, very popular. There are a few bacteria that are deficient in one of the enzymes, and so they are not able to do the Embden-Meyerhof-Parnas glycolysis. So we'll talk about that next. Okay. All right, so here's our Embden-Meyerhof glycolysis, and we're going to compare that to a pathway called the Entner-Doudoroff glycolytic pathway that only bacteria do, only some bacteria. Okay, so like I said, let's look first at this just glycolytic pathway here in the Meyerhof that has a little bit more detail drawn in. Oh, actually, let me step back just a second, because there was something that I forgot to say that I just wanted to say here because I had more room to write it. I talked about how a lot of the pathways of metabolism are actually amphibolic, okay, that they can actually proceed in either direction. Okay, this is one. We can actually, most organisms can actually, this pathway can actually go the other way, and we call that gluconeogenesis. Okay, if say an organism is not starting out with sugar, maybe it's metabolizing lipids instead, and it actually needs sugars to make some kind of cellular structure. Okay, then with just a few different enzymes and actually, of course, putting in ATP required, okay, then these reactions can go the other way and cells can actually make sugar. This fructose diphosphate, this is a precursor for peptidoglycan. Okay, so again, if cells don't have glucose around and they're using something else, they still need to make like, a cell wall or whatever. And so they can do that by a process known as gluconeogenesis. All right, just wanted to throw that in there. All right, so if we look at glucose just a little bit more closely, and I wrote in here, the net gains, that's what we talked about before. This step right here, this is the lysis step, that enzyme there that splits the fructose 1,6-diphosphate. Again, I said that molecule was not symmetric, so it doesn't split into two molecules that are exactly the same. It actually splits into this dihydroxyacetone phosphate, which actually is usually converted right back into glyceraldehyde phosphate. So that's why we don't usually make too big of a fuss about that. But this molecule becomes important if the cell is using some kind of lipids for energy. So just wanted to make note of that. We'll come back to that later. Again, this just kind of shows the intermediates, again, not to memorize. But that, like I said, there are some bacteria that are not able to do glycolysis because they're actually missing this enzyme that catalyzes this reaction right here. It's phosphofructokinase. Okay, so some bacteria cannot make that enzyme. So when they start going down this path and they get, oops, I think it's, yeah, sorry, it's this enzyme right there. I drew that in the wrong place. Okay, when they get to this molecule, fructose 6-phosphate, they can't go any further. So luckily those reactions are reversible. Sorry, it doesn't reverse that far. Okay, it would reverse back to glucose 6-phosphate, and it would enter in this Entner-Doudoroff pathway. So again, we call this an alternative to glycolysis. Only some bacteria can do it. Right? We're not doing this in our bodies, and we see there's a lot of similarities between Embden-Meyerhof glycolysis, but because of the deficiency in that enzyme, right away at the top here, you got to put in ATP, just like glycolysis. We talked about that kind of transport group translocation where the glucose is actually, a phosphate is added to the glucose, so it doesn't leave the cell right away again. Okay, that's what's happening here. The ATP is invested. And then right away here, there's a difference. We've got a different electron carrier in the Entner-Doudoroff pathway in ADPH. And remember that we said those electrons are going to power anabolic reactions. They're not going to electron transport chain to be converted into ATP. And then we get this keto-deoxyphosphogluconic acid, and right away, a pyruvate. Okay, so you see, actually, what we start with glucose, and we're actually going to end with 2 pyruvates, just like we did with glycolysis. But this first pyruvate comes off so quickly that no energy or electrons are gained from that. The second part of the pathway here is exactly like glycolysis. The other glyceraldehyde 3-phosphate we get an NADH, we get two ATPs out of that. Okay, so if we look at the net gains for the entner-Dodorov, we've got one NADPH. We get only one NADH. We still get two pyruvates, but we only get one ATP. Okay, so half the amount of ATP for entner-Dodorov than what we get for Embden-Meyerhof. Is that a problem for cells? Not really, because the bacteria that rely on entner-Dodorov, they are, I think, as far as I know, all of them are aerobic. Okay, so they're getting most of their ATP elsewhere. They're not getting most of their ATP from their glycolytic pathway. So, you know, 1, 2 enzyme doesn't make a difference because they're going to get 30 more somewhere else. Okay, so not a problem. And here's just an example. Don't memorize. We actually, the list of organisms that are able to do this pathway seems to kind of grow over time. Pseudomonas, that's an organism you're going to work with in lab. Enterococcus as well. E. coli will do the entner-Dodorov pathway if you grow it up on a certain medium. Give it certain nutrients that will do that pathway instead of regular glycolysis. Okay. There is one more alternative to glycolysis that we need to talk about called the pentose phosphate pathway. And this one looks complicated. We'll break it down. Sometimes it's referred to as the hexose monophosphate shunt. I won't ever call it that because that name just doesn't make as much sense to me. Pentose phosphate pathway makes more sense to me, and we'll talk about why that is. Okay. But when do organisms use this pathway when they have glucose instead of glycolysis? Well, they actually can be doing both pathways at once. But organisms are going to use this pathway when a cell's needs are more biosynthetic. So they need to make cellular structures rather than needing energy. Okay. And again, they can be doing pentose phosphate at the same time as their entner-Dodorov or at the same time as Embden-Meyerhof glycolysis, whichever it is that they do. All right, so let's see what they would gain from the pentose phosphate pathway. Right away here, you see some electrons gained with the electron carrier NADPH. So they get 2 NADPH. Remember, those electrons don't get taken to electron transport chain, but to power anabolic reactions. Question. Oh, so what's a biosynthetic reaction? So that's the cell, as we see, there's some precursor molecules that get gained from this pathway that are then used to make nucleic acids or used to make amino acids or something that the cell needs to, you know, make some protein or cell wall or DNA or RNA. Okay, good. So let's see. And then we have this 6-carbon compound here, glucose 6-phosphogluconic acid. We lose a carbon. We call that a decarboxylation when we lose a carbon dioxide. So now we have a 5-carbon compound, which this is complicated. It will either go one way or the other, not down both of these paths. But say we get ribose 5-phosphate. That's a really important precursor to make nucleotides. Okay, pentose sugars are what make up DNA and RNA. Okay, so it's written there kind of small, but ribose 5-phosphate And this is a precursor needed to make nucleotides. And we'll delve more into the structure of DNA probably next week. If that's not what the cell needs, then there can be some reorganizations here. And down here, this molecule, erythrose 4-phosphates, that's a precursor for particular kind of amino acids, ringed amino acids or aromatic amino acids. So erythrose 4-phosphates to make amino acids. And, you know, if the cell doesn't end up needing either one of those things, then it can make this glyceraldehyde 3-phosphate, or even end up here as another intermediates of glycolysis. So kind of whatever's left over can get just shunted into glycolysis and then still get a little ATP or maybe electrons out of glycolysis. But from pentose phosphate, if we're just staying right in this pathway, you'll notice that there's no ATP produced from this pathway itself. Okay, so no ATP from this pathway. There's a potential of getting some ATP if some of those compounds, again, kind of will go into one of the other steps of glycolysis. Okay, again, this is not just bacteria, but we do this. Lots of different organisms can do this, and even at the same time is what they're doing, glycolysis. Okay, all right, we're doing good. Stick with me here. I know wading through pathways takes some concentration. We're doing good. Okay, so we've come through some glycolytic pathway. Following how respiration will proceed, we've got this 3-carbon pyruvic acid. You know, whether again Semton-Meyerhoff or Ender-Dodroff, we get 2 3-carbon pyruvic acid. Next step is entering the Krebs cycle. And it turns out that only 2 carbon compounds can enter the Krebs cycle. So there has to be some kind of transition step to change pyruvic acid a little bit so it will be accepted into the Krebs cycle. So there's a decarboxylation, loses a carbon. So we get this 2-carbon acetate. It joins up with this carrier molecule called coenzyme A. And in the process, we actually get some electrons off of there. And what we end up with is acetyl coenzyme A. Okay, so sometimes this NADH is included in kind of sort of the totals from the Krebs cycle. Just kind of have to pay attention to how we're counting it. All right, so here's a overview of the Krebs cycle. And you notice that they labeled it citric acid cycle. Krebs cycle has many names. Krebs cycle, citric acid cycle, or even the tricarboxylic acid cycle, TCA cycle. Okay, that's all the same thing. And when we call it Krebs cycle, we give homage to the guy who elucidated the process. Citric acid cycle, it's named for one of the main first intermediates in the cycle. And sometimes we call it the tricarboxylic acid cycle because lots of the intermediates have 3 carboxyl groups on it. Okay, whatever you call it, this is taking place in the cytoplasm for prokaryotes and in the mitochondrial matrix of eukaryotes. So we don't really talk very much about mitochondrial structure, but they have kind of this inner membrane they have a plasma membrane, and then they have another membrane. You call it these Christie folds. So the mitochondrial matrix is going to be inside that inner membrane. Okay, so that's where this is taking place, as well as that transition step that we just talked about. So here's a 2-carbon acetyl-CoA kind of entering the citric acid cycle. It actually binds to a 4-carbon compound that our next slide shows us a little bit better. Net gains here. Let's go ahead and talk about that on this slide here. We get, let's count, we'll say per pyruvate. You could count that per glucose, and then you would double it, right? Because you get 2 pyruvates from each glucose. So per pyruvate, that's going to include that transition step. So here there's 3 NADHs. We're going to count that as 4 NADHs because there was 1 from that transition step. We get 1 FADH2 and 1 carbon dioxide from that transition step. And here we get 2. So let's count that as 3 carbon dioxides and 1 ATP. That's a substrate level phosphorylation. It's getting that phosphate. Actually, I think it's mostly from the solution, and whether it's GTP or ATP, we're not going to fuss about that. The GTP is often converted into ATP. Okay. Getting back to this 3 carbon dioxides. Okay, we had the 3-carbon pyruvic acid. We've lost all 3 of those carbons. So that's something to think about. Some organisms can't really afford to lose that much carbon. So we'll come back to that. This is another one of your book's pictures that just adds a little bit more detail to what's going on here. So I want to point out a few things. Okay, so we're starting here, this 2-carbon acetyl-CoA. That binds to this 4-carbon oxaloacetic acid and makes the 6-carbon citrate or citric acid. Okay, that's what we name the title, or the cycle, citric acid cycle. And then here's where we lose the carbon dioxide, we lose carbon dioxide here. The electrons come off at various places, either carried by NADH or FAD. Here's where we get the ATP down here. These little red squares that are circled around. That I don't want you to memorize that. I just kind of want you to appreciate that some of these intermediates, like in this case, alpha ketoglutarate, instead of continuing on in the Krebs cycle, that can be used as a precursor to make glutamate, which then can be used to make some amino acids or even nucleotides. Over here, this oxaloacetic can be used to make aspartate, which can be used to make nucleotides or amino acids, or especially sugars. So that's where, gluconeogenesis. So just to kind of appreciate, there's things coming and going, and it doesn't always follow in this neat little pathway that we think that it should, or the way we draw it out most of the time. Oh, what else did I want to say there? Okay. So there is one variation of the Krebs cycle that I want to finish up with here today. I said that those 3 carbons from pyruvic acid were lost. Okay, and some bacteria can't afford to do that because they need that carbon to make their structures. So there is a way that some bacteria can actually take this isocitrate and cut off the steps where they lost the carbon. Okay, so we call that And that's what I've got on our next slide here. We call that the glyoxylate cycle, or sometimes it's referred to as the glyoxylate shunt, because you're actually, again, cutting off. You can kind of see it in the background here, the other steps of the citric acid cycle. And again, let me talk through this, and then I'll talk, and then I'll, on the next slide, write down important keys I want you to take away. Okay, so this is something that bacteria can do, plants and fungi can do, but humans cannot. Okay, we cannot, we don't have the enzymes. There's 2 specific enzymes that are necessary for it to do this. Okay, so what happens here? Remember, we've got the 6-carbon citrate. This isocitrate is still 6 carbons. So instead of losing those carbons, we've got an enzyme here that cuts isocitrate into this 2-carbon glyoxylate and a 4-carbon succinate. Okay, so it's still 6 carbons. And then another acetyl-CoA, remember that was used in the transition step as well. Another acetyl-A, remember this is 2-carbon, binds with the glyoxylate and makes this 4-carbon malate. Okay, so in that way, they haven't lost those carbons. And this is, you're actually getting 2 copies of that malate, because you're getting the one that comes around and stays in the cycle, and then you're getting an extra one. Okay, so that extra malate can be used to synthesize whatever cellular structures that the cell needs. Okay, so you're losing, you don't get a substrate level phosphorylation, and you're losing 2 NADHs. You get, this is hard to read, but this is FAD here, and this is, you know, NADH here. You're still getting a little reducing power. Okay, so let me write down some bullet points here. What you should know about the glyoxylate shunt. So again, lots of organisms do it, not animals, okay, not humans. The main function here, so it bypasses the CO2 loss, so that carbon can be conserved. And used as a precursor. Often to make sugars, but it could be something else as well. And one other point is this allows organisms, especially bacteria. I'm not sure if other organisms can do this as well, but I know bacteria can, to use 2-carbon compounds, things like maybe acetate or ethanol, as an energy source. When glucose isn't present. Okay, so that's the main points of that glyoxylate shunt. There's evidence that some bacteria, it's absolutely necessary that they have that they're not pathogenic and they can't persist inside our cells unless they can do that. So that's why I'm mentioning it now, because we'll mention it at least briefly later. So, all right, so I filled your brains up. We'll finish out respiration next time and get into fermentation as well. Have a good weekend. Good week.
All the way to D-Meyer? Yeah.